It's interesting when you work on both sides of the streets to see how both sides of the street regard each other, academia and industry. And we heard this morning about a lot of obstacles and how they can be overcome. And I just want to point out that there's plenty of blame, if you wanted to be in that mode, to go around. For example, on the electronic health record, people like to point to the vendors, perhaps correctly, that the technology is vintage 1970s, 1980s for most of the vendors, and doesn't necessarily interoperate with the latest, greatest software, and nor was it necessarily engineered to do so. At the same time, though, we have to recognize that many large academic healthcare systems and commercial for-profit healthcare systems are not particularly interested in sharing data for a variety of commercial reasons. But they each often point at the other as the reason for why data sharing is not happening. Similarly, I see in many conversations that were, especially when I was younger, very confusing to me, when I was less cynical, about how industry doesn't know what it's doing when it's developing new therapies, or conversely, academia is purely theoretical, don't really know how to solve real-world problems. And from where I sit at this point, I've witnessed in my lifetime many, many times successful, groundbreaking collaborations between academia and industry. But often it's been because there have been a few people of goodwill, and there's the structures to help them bring them together have not been always the easiest. And it's for that reason that I thought we should have a panel on this, and I think we will start with no one better than Kathy Giusti, who not only, as we heard from Dean Daly, started on the industry side and then went into the nonprofit side, but bizarrely has done the full 180 and is now at the Harvard Business School leading the Kraft Precision Medicine Initiative, which precisely tries to do something in this space. And so I'd like to invite Kathy to give us her perspective.
Thanks, Zach. I'm so happy to be here and to share the models that we've developed and give you a bit of the story and our perspective on industry versus academia versus all of the above. I started in this whole field because I was diagnosed at the age of 37 with multiple myeloma. And I have to say, hearing Shirley speak earlier today meant so much to me because I remember feeling just like she did. I was 37, my daughter was 1 year old, and I was told that I had 3 years to live at best. And back then in multiple myeloma, there was nothing going on. I mean, I was working in the pharmaceutical industry, I studied the pipeline, and there was absolutely no reason for hope. But believing that if I just could jumpstart research and maybe live long enough that my daughter might remember me, I started the Multiple Myeloma Research Foundation as a nonprofit entity. And I remember studying the landscape and thinking, wow, there's hundreds of nonprofits out there, and less than 10%, it's even less than that, were raising more than $1 million. So how do you possibly start funding research if you can't even get these nonprofits off the ground to do that? So we jump-started at the MMRF model by funding the research to attract the scientists to the field. And once we started doing that, we realized that bringing the money together and bringing them together was great, but it was still really hard for them to collaborate. So we decided at the MMRF that we would also start building our own models, which became a collaborative tissue bank, where we asked everybody to bank tissue with us, follow the SOPs, and share the tissue and use it based off of prioritization. And today that tissue bank holds over 4,000 samples for the scientists to use. And then we started working with them in terms of building a genomic initiative. I was actually serving on the National Cancer Advisory Board when Eric Lander came in to present the TCGA. And at the time, I knew there was no way that the NCI was going to get to this little teeny tiny cancer called myeloma. So not realizing how important and big Eric Lander was, I just picked up the phone the next day and said to him, you know, what would it take for you to do this in multiple myeloma? And he said this number of samples, this access to myeloma expertise, and this amount of money. And we worked through an entire proposal and grant program pushing the road to work with TGen, and actually we're one of the first groups to sequence the myeloma genome. We then moved forward and said we had to build our own clinical network because we didn't have drugs in myeloma. So we started with 4 centers, and today we have 25 centers. We've now conducted over 75 phase 1 and 2 clinical trials in the field of myeloma. And to date, now 10 of those drugs have been approved in the field of multiple myeloma, and we have now tripled the lifespan of our patients from the very sad 3 years when I was diagnosed to 9 or 10 years today. And I think it really does show the impact of what happens when you bring your ecosystem together. It wasn't just the MMRF, it was the fact that we were constantly evaluating the landscape and writing the business plan to drive the ecosystem forward and aligning everybody around the incentives that would get them to play together. And you know, we'd always walk in and say, you know, an academic may be looking at it like this, but you know, we want Celgene to come in here and we want Millennium to come in. How do we do these trials? How do we all work together to get it done? And I will also tell you, access to money, access to patients, access to data drives a lot of really good behavior in an ecosystem. But with all of that progress that we made in myeloma, the one issue we had was it was still really hard to get data in the public domain. And it was still really hard to get the academic centers to share their data set. So over 5 years ago, we launched what we call the COMPASS Initiative, which was a $40 million initiative for the nonprofit to take on. And in the COMPASS study, we sequenced over 1,000 patients from that critical moment of diagnosis before they started treatment, and we continue to sequence them all the way through their journey every time they relapse. So we call the COMPASS data set a bit of the trifecta in that it is clinical, it is genomic, and it is longitudinal. The most important thing to know about the COMPASS data set, though, is that it is also housed in the public domain, and any scientist in the world has access to our COMPASS data set. So you would think that that would be an amazing thing. We built it also, just so you know, from a business perspective, as a pre-competitive consortium. So it was funded halfway through philanthropy and halfway through pharma. And what they got in exchange for the funding was just an early look at the data before everything went public. But my frustration most recently has been watching all the abstracts flow from the COMPASS data set. They're beautiful, they're eloquent, they're always done around the ASH meeting, so huge, lots of activity right around ASH. And yet I always wonder, is it really driving to the clinic? So an example would be of hypotheses that are great that have come out of that are transplant looks better than not. Triplets look better than doublets. African Americans look different than whites in our data set. But what do we do about all that? So due to the frustration around that, I had to sit with our ecosystem again and say, we've spent $40 million. What's it going to take to actually take these hypotheses and truly drive them to the clinic? And the actual answer became what we call the answer fund, which is we now have to be the trusted third party that takes the multitude of questions we have, and there are many of them coming out of COMPASS. The community has some, the academic centers have some, the payers have some, pharma has some, and most importantly, the patients have many. How do we prioritize the questions, and then how do we fund those questions? So ironically, here we've spent $40 million to get the playing of everybody, and to get data in the public domain, and now we have to execute more funding to actually identify the questions and then provide the resources, the staffing, the incentive by which people will answer very specific questions. And Zach and I were having this conversation yesterday, which is if you prioritize the questions and you actually send them out to your entire database and you start to crowd them, what are the most important ones, what do we want to do? People will start applying because, of course, there's money and there's data. And as I said, when you have money and data, you get people to play, and you start to understand the landscape better. And you start to know you have the alignment of the willing, right? So you know who wants to share their data, and they have to share their data to get the funding. And so you have to keep building on those ideas, and again, it becomes a best practice model in business of how did we get this done? It seems like such an obvious thing to do. It has literally taken our office 6 months to actually make this all happen. So that's the space that we're moving forward at the MMRF to keep answering the questions that our patients deserve to have answered. And just to give you a quick signal of that, if we know that high risk is one of those areas, and we want to build that out and say, why did we find that translocation of 414 patients, 2/3 do well and 1/3 die right away? But those are only 15% of our patient population. We need lots of data sets to come together around that question and help us to answer it. So that would be a very well-framed question. Another would be, that's broader, should we be on maintenance therapy or should we not? You know, myeloma patients now live a long time. If you're in a stringent CR, do you do it or do you not? That's more of a payer question right now than a pharma question. So trying to get all the alignment is critically important, which makes me fast forward to just inform you that my job half-time is at the MMRF, moving this model forward that brings all players in the ecosystem together, but simultaneously I'm half-time at Harvard Business School, because the frustration is when you have, and Zach said this, when you have models that are working, you need to be sharing them real-time with other cancers. So at HBS, we now work in 4 different work streams across all cancers. The first work stream is direct-to-patient, where we work with all of them to understand how do you bring in Uber and Rent the Runway and Amazon to teach us how to do data sets better, how to ask questions better, how to engage patients much better. The second is on data and analytics. The third is on clinical trials and how you build out platform studies. And the fourth is on investment models. But everything we do at the business school is trying to bring together the best-in-class people, only the disruptors, people that really are tired of the status quo and will be brutally honest in all of their discussions. And we keep these solutions teams together for the long haul to make sure that we're identifying new solutions and disseminating them through the entire Harvard infrastructure, which has been a real gift to all of us to work with. And the joy of it, I think, really is the fact that the Kraft family, it's endowed by Robert and Jonathan Kraft, who you all know from the Patriots, but their passion is to make sure that the business of science moves forward quickly and that we understand the role of business and incentives to make sure science and technology explode as they should, and nothing's closer aligned to the way that I feel too. So thank you.
So next we have a twofer. Do I see you? Oh, yes, I do see you. So I have a long-standing friendship with 2 colleagues. One who I met through another sociologist, through a sociologist at the University of Pennsylvania, and one who I met when he was fresh out of medical school, having previously graduated from MIT, and who wanted to make a difference in health IT. And it turns out they have both come together working with our department on a project that I think is emblematic of how in precision medicine we could work with some of the larger commercial companies. And I have to say, and I hope they will communicate that in their joint talk, that we've been pleasantly surprised that some of the entities that we believed not that interested in sharing data, in fact, in the right context, are extremely amenable to it. So without further ado, I'll introduce first Josh Mandel, followed by David Kreda. Thank you very much.
Thanks. So I'm excited to get to talk with you briefly about a project that we've been working on called Seeq for Science. So Seeq as in synchronize your data. And the value proposition here is really putting consumers in control of how they share their electronic health record data and making it easy for consumers to decide to share their data with a research study and then at the click of a button, make that happen. And our goal has really been to do this using standard consumer web technology. So if you've ever shared data from Facebook or your Google account or your Twitter account with a third-party app, that's the kind of technology that we're really focused on here. And the goal being that any researcher or any research study can build an app that asks for permission. Now, a patient doesn't have to grant it, but they have the opportunity to do that. So this is the kind of workflow that we've been focused on, and I want to describe two interesting things about the project. So the first is that the project was funded as an academic research grant here at the Department of Biomedical Informatics. And I started when I was working at the department, working with a small team there on building some technology infrastructure to help. But the interesting thing is that it wasn't really a project for us to develop new technology. Our mandate was to work with the community of electronic health record vendors. So big companies like Cerner and Epic and Allscripts and eClinicalWorks that among them together have a majority of the market share for hospital systems and a large number of outpatient or ambulatory systems as well. So our mandate was to try to figure out how to work with them to get support for this kind of technology in a consistent way built into the various patient portal products that they produce. And it's been an interesting journey. The second sort of detail that I want to point out is that those vendors who have been working with us aren't being funded to do this work. They've effectively volunteered to participate. And their motivation for that is interesting. One piece of it has to do with our regulatory environment. So today we have, let's say the last 7 years or so, a national program of incentivizing doctors and hospitals to make meaningful use of electronic health record systems, and there's many details about what goes along with that. But one of the details is that you need to use a certified health record system in order to be eligible for those incentive payments. And so vendors don't have to get their product certified, but it's generally in their interest to do so, and they all do, because it's a major selling point for their product. So there's been an interesting development in the regulations, which is to say that coming online next year, so late 2017 or early 2018, as part of Meaningful Use Stage 3, providers need to give patients a way not just to see their data online in a portal, but to share those data with an app of a patient's choice. So it gives patients not just the right, which they've always had, but the technical ability to make that sharing decision and feed data into an app. And so this, to Noga's point from earlier today, when she was making the distinction between stuff that works on the ground right now and those beautiful APIs that are coming in just 12 to 18 months, I'm here to tell you about those beautiful APIs. The fun thing is they're actually working in production today at a number of these vendor sites, and we expect that those numbers are going to grow over the next 12 to 18 months, but already they're out there, just not evenly distributed. So I'm going to pass over to David Kreda to tell you a bit about the way that we've been building out the infrastructure and working with the vendors to make this happen.
So good afternoon. I'm actually more of the business person, at least for purposes of today's chat. And I wanted to give you a vision of what this endeavor would look like if you thought about playing to succeed and to win. So this particular technical platform is a platform that actually individual vendors are enabling through their patient portals. And initially, they're provider EHRs and a few EHR aggregators. But we believe that other systems, payer systems, imaging systems, lab systems, and they could be including genomic lab systems, could participate so long as they also expose a patient portal where individuals can see data or have data moved. On the receiving end, we are starting with the All of Us Research program as it were the charter research app that's going to consume data, but any number of other research programs could build apps and use the software. Indeed, that's the intent behind this from the perspective of the NIH. What Josh did not mention, but is worth commenting, is that the same API and approach can be used also by consumer health apps. So this has essentially horizontal capacity to impact all along the patient-centered domain. Now, as I mentioned business, I think it's always appropriate to kind of bring out an S-curve and talk about the potential dynamics. And what I would point out is that we have our first movers, which constitute ultimately over the next 18 months more than 50% of the footprint of hospitals and clinics in terms of having latent software in their portals for individuals to donate data. And what we will be doing from the point after the initial pilots is that there will be more and more EHR systems that have this upgraded software. And we expect, because we've seen some examples of people already preparing to write research apps to use the same technology to get clinical data through this mechanism. What we're actually hoping is that as we get also additional types of vendors, we're going to weave this technology into the health IT infrastructure of our country, fundamentally changing the economics of doing donations. And that, by the way, can include delivering data not just to research studies, but to intermediaries that, for example, construct longitudinal medical records like Noga's company. Now, there are a few things I would mention. There are many dominoes in this, but I want to just bring up two, which I think are significant factors for achieving success. One of them is that the consumer engagement activity in apps is a new skill set. And in fact, researchers in building these participant-facing apps are going to have to figure out how to engage individuals to visit these apps, use them to share data, not collect so much information up front that they essentially abuse the potential participant and they drop out. All those skill sets are going to have to be acquired, probably with some trial and error and probably through some consolidation with business partners. The other thing is that, as Josh mentioned, the meaningful use stage 3 requirement about exposing an API, that particular requirement, if it's just slightly tweaked, could become characteristic of the entire health IT environment, where that is a requirement for any clinical or clinical-grade system that has data about an individual. With that slight tweak, very focused, it would be possible to endow the entire industry with a motive, in addition to potentially money, to expose this API and make this ubiquitous. One last thing I wanted to state is that if one were thinking about the value proposition for this type of technology, the way to think about it is, how does it change unit economics? How does it change behaviors? How does it make the essential delivery of a service faster and better? And what we'd like to sort of say about this is that it's a quick thing for an individual to say, I want to share my data. It could be done with under 2 minutes from the time you actually go to the portal login and say, I approve it. It's cheap, but that would mean that the incremental cost for delivering data from an EHR to an app is probably more like $0.01 than $100. Obviously, at those orders of magnitude, you'd expect a significant change in the way people think about things. Its ubiquitousness, well, I demonstrated that if an S-curve comes to pass, it will be ubiquitous, and of course that changes the way what people think. They just assume it's a commodity, it's the standard. Its transparency comes from the fact that as an individual, when you say, I want to donate data, right, you know what you're doing and what you're sharing. It's quite explicit. It's deterministic. This is actually one of the most difficult challenges when people are assembling medical records, which is, I have different patient IDs. How do I know if it's the same individual? But this is driven by the individual who goes to her own patient portals and says, those are my data, and sends it. So you essentially eliminate that type of source of confusion, even danger. The data that are coming out initially are structured data, and these are the common clinical data set. Medications, allergies, problems, the type of things that actually constitute a core of data for many studies. It's almost sufficient to do all kinds of work. The only part that our initial service doesn't really deliver is, in the main, is free text data. And that's only because I feel that that was the one thing that was left out of that meaningful use stage 3 requirement. Add that one thing, and you can start having these data delivered for NLP and the type of computational phenotyping that allows you to amplify and curate the structured data in a manner that would make for better science. And I think overall, as we've tried to convey, this is part of the infrastructure of delivering content, medical data into research and into also consumer health apps. It has a fundamental possibility of changing the entire economics and changing, therefore, a lot of behavior for research and for healthcare. Thanks.
So one of the scariest stories I ever read was about an academic who was doing extremely well, who was extremely valued by his institution, and then just decided that he didn't need that institution necessarily, and in fact might be even more successful outside that institution. And that person is Ethan Perlstein. And so I was been very, very impressed and scared because it's really a new model of the relationship between industry and academia. And because he's not dead yet, I think we actually have room to believe that this is going to be a successful model. So Ethan, please let us know. Thank you.
Thank you, Zach. I am indeed not dead yet. This is actually a nice welcome, homecoming for me. I was a grad student across the river, Department of Molecular Cell Biology, about 10 years ago. So it's nice to be back and talking to sort of a lot of what I see in the audience are sort of the next generation. So if I had to describe, so I'm the founder and CEO of Perlara. We're a South San Francisco-based biotech startup. If I had to describe Perlara's value proposition in a phrase, it would be economies of rare. What do I mean by economies of rare? Obviously, that's a reference to economies of scale, but it's really a re-articulation of the nugget of wisdom that Noga passed on to you this morning, which is do things that don't scale, but apply to rare disease communities. And you might think, based on what Matt Might said, that aren't rare disease communities, especially ultra-rare disease communities, where there might be fewer than 60 people on Earth, isn't that the definition of unscalable? But I think all of the equations for scaling are missing a term, which is motivation, and that the basic unit here that we're talking about is the highly motivated family, and they can move mountains, as Matt Might and every other parent with a child with rare disease testifies to by their actions. So what does that actually mean in practice? What do we actually do? So we really are doing two things. We're innovating on our scientific model and are innovating on the business model. So on the scientific side, we're using model organisms like yeast and worms and flies and fish to model rare diseases. And essentially taking this phenotypic screening approach, but instead of using cells, we do actually use human cells. We're really primarily driving drug discovery through whole animals. So that, 99% of pharma is really based on either target-based screens, in vitro, or cell-based screens. So I can talk to you why why we're contrarian here, but I think actually the mainstream is coming back our way. So phenotypic screens with whole animals, that's our scientific innovation. The business model innovation is really partnering with the highly motivated families out there with children with rare diseases, and not exclusively just those with children, but also the advocates themselves. These groups are coming to us and kickstarting drug discovery for their disease because they're being ignored and neglected by everybody else in the world. They're too tiny for pharma, and you really can't do this kind of integrated translational research in a bunch of scattered academic labs quickly and efficiently. But I want to just take a little bit of time just to talk about my story, because my story is really about coming out of academia and going into startup land. And it's not the first. I'm sure I'm not the first person to do this, but I think there are some elements to my tale that hopefully will apply to others and are kind of characteristic, I think, of the times we live in. So I left academia at the end of 2012 because of the postdocalypse. If you don't know what the postdocalypse is, then you're probably not a grad student or postdoc. But if you are, you know exactly what I'm talking about. And briefly, it really just relates to the fact that there are way too many postdocs being produced for the number of academic jobs that open up every year. And a postdoc is really meant to be training for an academic job, and so there's a problem because there's 7 postdocs for every one opening. So I realized that, and the writing's been on the wall. I think the writing was on the wall even before that, but now we live in times 5 years later that seem like even worse than they were back then. But for me, leaving academia was actually plan B. That was not what I wanted to do ever since I was younger. But I was forced by circumstances to come up with a plan B. And in fact, Twitter played a huge role in helping me reestablish my professional identity, meet all kinds of people like VCs, experts in things I didn't know anything about. But most importantly, it was where I found rare disease communities. They were able to go to platforms like Facebook and Twitter and start to make themselves heard, make a lot of noise, right? Because 57 NGLI1 patients in the world right now is hard to hear that in all the din. And so Twitter is this amazing place I discovered. If you can avoid the trolls and some of the other downsides, it's an amazing place for communities to essentially take control of their destiny. And now with Facebook allowing people to fundraise off of their profile page, I think this is sort of just the tip of the spear in terms of how rare disease patient advocacy has really come onto the stage in the last few years. And then really the real pivotal turning point for me personally was attending Rare Disease Day in 2013 at NIH. And that's when I really first met families and real people with rare disease. Before it had just been this abstract thing. I taught Mendelian genetics and in grad school and in my postdoc, and it was all it was nothing personal. And finally I met human beings who were affected. And then started to ask some questions like, hey, are people making simple animal models? Because I had done a grad school and postdoc looking at yeast as a model for pharmacology, and started asking people, are people using animal models? And everyone just said, you mean a mouse model? I said, no, there's other animals. Look at Noah's Ark. Look at the smaller things that you don't see, like the worms and the flies, but they're there too. And convinced myself through that kind of diligence that people really weren't using model organisms as I thought they should be in in drug discovery, and thought, hey, maybe there's actually an opportunity here. It was also by interacting with folks on Twitter that I came across this idea, and in fact my brother also played a big role in introducing me to this concept of a public benefit corporation. And that's this idea that if you have a C Corp here and a 501c3 here, PBC is here. So you're still for profit, but you get to enshrine in your articles of incorporation a social and environmental mission. And so technically this is hopefully a way that the industry, I think pharma as a whole particularly, can start to rehabilitate its image and start to walk the walk. You hear a lot of talk about patient engagement and patients first, and yet at the same time the cognitive dissonance of drug prices going up, pipeline drying up. So something's got to change, and we think the fundamental way to do that is to write into your DNA that you're a company that's balancing, maximizing profit with this mission of helping patients that are being neglected. So by early 2014, Pearlstein Lab was born. So it was originally called Pearlstein Lab. I wasn't very creative in coming up with a name, and also wanted to signal a bit that we have some academic roots. We became Perlara last year, but essentially the company was born as a hybrid of a sort of academic lab and a startup. Basically an academic lab with like, a lot of deadlines that you have to really meet, or the company goes away. And so that was really the first year of the company was 2015, getting our bearings and actually trying to get noticed. The patient groups were noticing us, but we weren't quite ready to work with them. What we needed was validation from a traditional pharma player to say that our platform, which is a little bit innovative scientifically, the business model is a little bit different. We needed that stamp of approval, so we got a partnership with Novartis last October. I was actually just there the last 2 days for meetings on our collaboration, partnering with 3 different diseases with them in the lysosomal disease space and making a lot of great progress. And I'll just wrap up by saying that, as I said at the beginning, one of the major ways we've tweaked the business model is to essentially say, let patients kickstart their own drug discovery when no one else is doing it for them. And we call this model PearlQuest. And so far we've launched 3 of these collaborations. We're working at NGLI1 and working with 2 other groups. One of those groups actually was referred by Matt Might. So the community there is, obviously they talk to each other. But we're really excited about these 3 PearlQuests that we've launched, and we've got many others that we'd like to launch. A lot of families looking to do this, and of course there's thousands of monogenic diseases out there. So we're really excited about this idea of sort of rebuilding the public benefit part of pharma, sort of from the bottom up, directly hand in hand with patient groups. Thanks.
Did I hear you say Pearl Quest? Correct. So it'll be like Pearl Star, will be all your products? Maybe. All right. So we've heard a lot about how patients should get access to all their genomic data and how that should be shared. And so I was quite struck that this was not a futuristic plan, but it was actually the plan of someone who I respected immensely because of his leadership in population genetics. But Joe Pickrell had actually decided to turn that into a quasi-business, which may actually be a business now. And so I was also struck by the fact that from day one, he didn't have the promise of an API, but an actual API built into the business plan. So I have a strong feeling of the future is now when I talk to Joe, and forgive me, Joe, if I screw this up, but I believe that when I first talked to you, you were fully at the New York Genome Center, and now you're fully at Seeq.io. So I take full credit for that. All right, thank you very much.
Thanks a lot for having me out. It's really a pleasure to be here. And so what I want to tell you all about today is exactly that. So about 1 week ago, I was a professor at the New York Genome Center in New York and in Columbia. And today I am the CEO of a company that I want to tell you about. And I want to tell you about how this project started, this project called Seeq. If I can grab this. slides, maybe. And tell you about the project and how we decided to make this into a company. And so the story starts a couple of years ago when I had just started my lab at the New York Genome Center. I'm a statistical geneticist and population geneticist. I actually did my postdoc upstairs in this very building. And I was starting a lab, and like everyone else has already mentioned, what you want as a statistician is data. That's what I needed. In order to make any progress in human genomics, we don't need data from 50 people or 5,000 people. We need data from hundreds of thousands or millions of people. And I looked around me, and my lab was 3 people, and some computers, and no actual lab. And so I thought we had a problem. And so I sat down with my lab, and we decided that we needed to reinvent a few of the key parts of a research study. So first, we went after sequencing. And so this needed to be cheap. We actually had no budget either. So we had no money and no lab and no people. And so the first thing we went after was sequencing, and we developed a really inexpensive sequencing technology. It's not quite clinical grade. Let's call it a research quality genome sequence, which you can go, and I think our price now, is $60. You can go ahead and download the app and order this right now. And then the second thing we decided to go after was how to actually get it into people's hands. Like the standard way to do this, of course, is to go to a medical center and get data indirectly through the medical system. We decided to do this differently, go directly to people. So you can go and download this and participate in our research. And in order to incentivize people to participate in our research, of course, we had to give something back, right? And so using our new sequencing technology, we developed algorithms and approaches that led us to a couple of things. So you spit in a tube, we sequence your DNA. We can do things like ancestry analysis. And since we're actually sequencing your saliva, we get the microbiome of your mouth as well, and some people find that interesting. And so the idea was we could make this sort of transaction. People are interested in their ancestry and microbiome. We're interested in research. Let's make this work. And so we launched this in August, like Zach mentioned, and it turns out a lot of the interest we got was not necessarily we got interest from people who wanted to participate. We also got a lot of interest from other research groups, small technology companies, small biotechs, because they want to do the same sort of thing that we want to do. They know they need to get to hundreds of thousands or millions of people if they want to make progress with the data, but they don't exactly know how to do it. And so we built out our API in such a way that individuals, participants, can share their data with anybody. You have to sort of sign up on our site, and people can share their data with you. But also, by interacting with our API from a computer or from your small lab, you can start shipping saliva collection kits around the country. They come back to us. We do the sample processing. We do the sequencing, and we ship you the data. And so you can start building your 100,000-person, million-person biobank basically with an internet connection. And that's sort of the value proposition of the company. And so why do this as a company, right? So this started as an academic project. There are a couple reasons. One is sort of instead of calling a business model, let's call it a sustainability model. How can you expect to do something like this long term? Like I'm in this for hopefully decades of my life, right? And the sustainability model in the academic setting, of course, is you write grants, you spend them, you write for more grants, you spend them, and the sustainability here it seems pretty precarious. Whereas as a company, they usually raise some money, spend it, raise some money, spend it, and hopefully then it becomes self-sustaining, and that's of course the goal. But the second thing, of course, is that as soon as we launched this, everyone just assumed we were a company anyways. And people write us at 3 o'clock in the morning asking us to fix their issues immediately. And Google is free, so why shouldn't why shouldn't we respond immediately as well, right? And it's a fair point. Google, of course, has thousands. I don't even know how many employees they have, and we have like 2. But there's a certain level of expectation in the consumer that is very difficult to meet in an academic setting, and basically, as far as I'm concerned, like the top people for doing that are working in industry. the sort of consumer engagement type approach. And so basically this had to be a company for those particular reasons. And to close, Zach had sort of asked, why is there sort of mistrust across academia and industry? And it seems to me it's almost like talking about politics and religion, right? People already know what they believe ahead of time, and there's no way around it. And so what I'd encourage you all, instead of, it seems, I guess, backing up, it seems what people do is they decide whether they're an academic or they work in industry, and then they decide what project they want to do. Whereas I encourage you to sort of flip it around, like decide what you want to do, and then take the route that is the most advantageous to actually get you where you need to be in the end, whether that's academia or whether that's industry. Each has their pluses and minuses depending on where you want to end up. With that, thanks a lot.
And sometimes getting a kick in the face apparently is the fastest way to convince someone to bridge that divide between academia and industry. And so I would like to invite Dr. Giselle Sholler, where is she? Oh, there she is, Giselle, to come and explain to us what I just meant by that. Thank you very much.
Sure. Thank you, Zach, thank you for having me here today. It's really an honor to be able to be with all of you with such incredible stories. And I'm happy to share a little bit about our story. So my name is Giselle Sholler. I am a pediatric oncologist at Helen DeVos Children's Hospital. There I'm the director of pediatric oncology research and their innovative therapeutics clinic. My story started about 10 years ago in doing pediatric oncology, focusing on neuroblastoma and essentially doing the same thing for children and getting the same outcome and going to too many funerals with little caskets. And so really a group of oncologists and parents decided we really needed to do something better for these kids and needed to find new options and therapies for them. So we came together. It was really a grassroots effort of, at that time, it was 5 oncologists, 5 different hospitals, and a group of parents who then started their own foundations and were able to fund the work that we were doing to get new clinical trials available for our kids. And after 10 years worth of growth, over the last 10 years, we've had 18 clinical trials and have grown now to be over 40 children's hospitals, most in the US, but we also have a hospital in Lebanon, in France, and we are in the process of opening 6 hospitals in Canada as well. So it's really grown. Actually, just yesterday, we changed our name. We were called the Neuroblastoma and Medulloblastoma Translational Research Consortium, or NMTRC for short. Still a mouthful, and no one actually knew what it meant. So yesterday, the NMTRC became Beat Childhood Cancer. Much easier and clear what our mission and our goal is. So we have 2 main arms. One is really the Dell Precision Medicine program, and the other is really a drug development program. I'll save that second one, which relates to Zach, in just a minute. I just want to introduce both programs. We've had to work very closely with industry. We are academics, and I think one of the questions Zach had asked was, should we start in academics or industry? And I feel that all our projects starting in academics has given us a nimbleness to be able to move rather quickly. I think it takes away any conflict of interest questions. But that I think to get to the finish line, it is important to work with industry and for accessibility to a larger population. And so one of our key to collaborating has been with industry, has been really aligned expectations. And that's where we found our relationships just to blossom and grow really, really well. And that this often means that patient outcomes have to be as equally or more important than the financial outcomes. And that's where sometimes there has been some challenges, which I'll talk about. But essentially, our Dell Precision Medicine program is a program we started essentially 5 years ago. Was when Dell Computers really became partners through TGen and working with Dr. Jeff Trent to really bring precision medicine to pediatric cancer. The year before that, we wanted to do a study in pediatric cancer for our neuroblastoma patients where we would look at the sequencing of the patient's tumor and see if we could identify targets to treat the patients. This was interesting. This is only 6 years ago, but the IRB allowed us to study 5 patients, incurable pediatric children with neuroblastoma, incurable at the time, to do the sequencing, to host the molecular tumor board at the time. That was the first proposal for that. Come up with a treatment plan. We were screening over 200 different drugs that already had dosing in pediatrics. We were going to combine 4 different drugs together, but we were not allowed to treat the patients. They wanted to see what what the tumor board was going to come up with by looking at genomic information. So 5 funerals later, and they looked at which was incredibly frustrating, as you can imagine as an oncologist, that the treatment plans that the tumor board from multiple different institutions came up with were all very reasonable treatment plans for these patients. So then we moved forward. Dell came in with the funding, and we were able to initiate the first trial using DNA sequencing. At that time, we were doing RNA expression profile. Over the last 5 years, it has changed dramatically. Now we have, again, multiple hospitals. And we are doing full RNA sequencing and DNA exomes for every patient, using a great bioinformatics team to come up with treatment plans looking not only at the mutations and fusions, but as well as the biology and the over and under expression of pathways, and putting together, again, up to 4 different drugs together in a treatment plan for the patients. And this relationship with Dell has been just nothing short of miraculous, really allowing the physicians and researchers to write the clinical trial to address our patients' need and looking at genomic information, while Dell, in partnership, has really increased our compute power and transfer and storage of large amounts of data, shortening the time from sequencing from 2 months to 2 weeks, really being able to do this in real time for our patients. We initially, in that first trial, required an investigational device from the FDA as well, because of the risk. We've already, clearly everyone's moved on from that. And instead of just being able to treat incurable patients, this last year now we're able to use this in neuroblastoma patients at diagnosis. So at diagnosis now, the patients are receiving the full analysis, and we're able to add, what we're doing is adding one targeted agent that's specific for that patient onto the standard of care treatment to see if we can improve outcomes and not wait until they're relapsed and incurable at that point. We've also incorporated in the last year a patients like me analysis, because we have over 200 children that we've done this with over the last 5 years. Our bioinformaticians will look and cluster the patients together to see what patients are closest genomically to that patient. We're doing it in all pediatric cancers now, not just neuroblastoma. And then what were those patients treated with, because we have the clinical information as well, and how did they do? And are those same drugs coming up on the report for this patient, and should we be using those? So really, the patients like me has been a great addition. This year, we'll be incorporating proteomics and epigenetics into the tumor board. So I'm looking forward to seeing how that goes. For each of the patients also, we have created cell lines and are starting to do the high throughput drug screening with NESWIC and PRESWIC and NCI libraries. And so at some point, we'd like to see that incorporated into the tumor board as well. It's a very exciting time, and it's exciting to hear how many other people here are doing the same thing. One of the other things we've worked on in the last couple of years has been working with insurance companies, and Priority Health in Michigan has approved DNA exomes and RNA-seq for pediatric cancers now, having gone through many cases with them to talk about really looking at the previous relapse therapies the patients were getting, which most of was high-dose chemotherapies with a lot of toxicities at inpatient time, versus the outcomes that we're seeing with targeted therapy and precision medicine, which is mostly outpatient, fewer toxicities, less costly to the insurance company than the standard therapy they were getting before. So that's another exciting change that we've seen. So this highlights really a mutually beneficial relationship between industry and academics. And really seeing kids graduate from hospice is one of the most wonderful things, and we have. And so that's just so motivating for us as a team together. So the other arm of our world is drug development. And over the last year, one of the drugs that we've been working on has gotten a lot of attention, and it's a drug called DFMO. It's a repurposed drug. It's actually used for African sleeping sickness by the World Health Organization, but not approved in the US. We've been repurposing it for use in neuroblastoma. And I'm not going to go into the biology of all that because that's not what we're here for today, but essentially, a few years ago, we as an oncology group in the consortium, the parents and advocates, wrote first the phase 1 and phase 2 clinical trial using DFMO in neuroblastoma. We hold the IND, done all the regulatory work, the data collection, the clinical monitoring, and DSMBs, but we needed to purchase the drug from a drug company. They were developing it in colon cancer, and so they didn't have drug available for us to donate, so we were actually paying for the drug for our clinical trial. But they, in the contract, had regulatory rights to move this forward for drug approval. We agreed because we assumed that we wanted to work with them to go forward for drug approval. That's the ultimate goal. 2 years. The study is essentially using this medication at the end of upfront therapy for 2 years to prevent relapse, because 40% of kids with neuroblastoma relapse, and when they relapse, there's no cure. So we wanted to see if we could prevent that relapse. 2 years into the study, the trial was going very well, too well. The drug company decided at that point that they wanted to stop selling the drug to us because they could do this better, and they wanted to do a placebo-controlled randomized study, like the adult world. So half of the kids would get placebo, half the kids would get drug. But the concern at this point, as our group came together, our statisticians were like, whoa, you're only halfway through this study. If you stop the study now, we won't have the power to know that these results are true. And what we're seeing is kids aren't relapsing. This is great. The parents who are on the study didn't want the drug to be taken away. The parents who were planning to come to the study, some of them coming from England and Ireland, were afraid they weren't going to get to go on the study. The company told us straight out, if we keep your trial open, no one will enroll in our study. We have to close your study because everyone got drug. I think there's just one of the misunderstandings, misconception between academics and industry is that academia can't run a rigorous FDA approvable trial, that the industry needs to take over, and it needs to be done in a certain way. And sometimes in pediatrics, the adult path doesn't always work. We actually talked about with our statisticians, the trial they were proposing would take at least 10 to 15 years to enroll, and that would be if oncologists would put their patients on that. None of the oncologists in our group would want to put our patient on that study. None of our parents would want to enroll in that study. So it was flawed to not succeed from the beginning. So as we were brainstorming, two of our parent advocates, Meryl Whitmer and Pat Lacey, who's straight over there, decided, well, we're just going to have to open our own drug company. I know, funny, right? They did. In less than 3 months. Where there's a will, there's a way, and it's amazing to me how parents and patients can do the impossible, and they did. They imported the API, and they formulated the tablet, they had it made, they had a distributor, they worked with regulatory people in DC, and in a matter of 3 months, they had a virtual pharmaceutical company, Kids Cure Pharma, or KC Pharma for short. Their goal, and what's even more exciting is their goal really is to have a nonprofit pharmaceutical company, which is a totally different model. That any profits that would come from the sale of that drug would go right back into pediatric cancer research and fund more research. We're not there yet. We still have a long way to go. But we then had to work with, we had 3 months. We had to work with the FDA to switch drug supply mid-study. So on the oncologist side, the FDA was absolutely amazing to work with. And we were able to do all the regulatory paperwork, which was a lot, and a lot of sleepless nights to see if this was really going to happen. But no patient went without drug. All the patients that wanted to enroll in the study did. The study results are incredible. We're just publishing them now from the first study. Since then, we've gone to the FDA. Our second confirmatory study is ongoing right now, and this has just been an incredible learning experience for me, and I think we're still learning in terms of running industry on our own, how to continue to move this forward. But I'll stop there, and I think it's wonderful to see that as academics with power and passion and the parent advocates together that we can make a difference.
As we're gathering, it strikes me that this is not a panel I don't think we could have assembled 5 years ago. Things are really changing, and I think the culture of jumping that bridge between academia and industry has shifted. Honestly, it shifted a lot earlier on the West Coast, but it's definitely happened here as well. And I want you to start thinking about questions, because I take the blame for the fact that I really packed this panel so that we only have 15 minutes left. So if you're going to ask questions, start thinking about them, and because after my opening question, I'll leave it open to you, but we won't have that much time. So I'm going to, since I always like copying my betters, I'll just borrow this great question that we had from David Shaywitz. And I'll ask each one of you, pick the one thing. If there was one thing you could do to change, that could be changed, and short of, I mean, all of you are doing miraculous things, so short of miracle is fine, but just don't go full miracle on me. Short of a miracle, what is the one thing that that if changed, would make that industrial and academic collaboration work better? And I'll start with Kathy, and we'll work our way across.
I would say the one thing that we would love to change is instant knowledge amongst the patient community that their data actually is on the critical path to finding cures. I think that patients don't realize that it is their data, it is their tissue, and if they had more of an uproar around themselves, that it wouldn't matter if it's industry versus academia, because the patient would be driving it. What we have found overall is many, many patients believe that as soon as you go to your doctor, that data will be shared. And many are told, yes, we are sharing it, and it's true. You may have a center sharing it with another center, but that's not exactly building the critical mass of data that we need, especially in some of the more uncommon or high-risk diseases. So I do believe that there's going to be some kind of a movement. We can sense it even in this room today, that there's so much more knowledge now with social media for the patients, but I wish it could happen like this so we could make it happen faster.
Amen. David.
So uncharacteristically, I'll confine myself just to health IT and regulation. In the 6 or 7 years in which I've been involved in some way and therefore seen the impact of regulation to improve health IT, I would say generally it's not accomplished its objective. For our purposes, if we treated interoperability as this type of commoditization that we all seek so that we can eliminate that as being a barrier for moving data, I would say that what we most need is for the regulatory apparatus to declare it nearly job number one and establish a simple and effective way to have a minimum viable regulatory regime that causes industry not to be highly creative in how they build APIs, but actually very disciplined in saying, if we do this, we'll be able to move data around. I think that would do a great deal to liberate energies into other higher value activities.
And maybe I'll just put a fine point on David's comment about regulation. So in our interactions with EHR vendors, we've seen that many of these companies are distracted, focusing on trying to meet lots of regulatory objectives, and it gets in the way of their product focus, it gets in the way of their actually being responsive to customers and patients. It's a real distraction. That said, some of the regulatory specifications, specifically around data and patient access, so that the consumer right to share your own data, have been incredibly helpful in focusing those vendors. So I would change something very small. I would ask the Centers for Medicare and Medicaid Services to focus on that definition of our common clinical data set today. It's the stuff that patients can share easily in an electronic way and expand it. Today it's basically structured clinical data. It would be an easy thing to expand that list to include free text clinical notes and to include imaging data. So any kind of imaging studies that have been done for a patient, make those things part of the data set as well, so that when patients make that decision to share electronically, all of those kinds of information can come along for the ride and not just blood pressures and lab tests.
I think one of the bigger problems is allopatric speciation, and what that means is that you've got the scientists in the industry and the scientists in academia, and they're no longer mixing. And I think the one easy fix would be add another rotation to grad students' rotation and call it a biotech internship or something, a flex rotation.
Great idea.
So I have very few complaints, but one opportunity, let's say, that I saw is something along the lines of the regulatory issues is around institutional review boards and opportunities for mobile research. And we had a hard time eventually. I mean, they're institutional review boards and they're institutional review boards, people that worked with us to get informed consent via mobile interfaces and things like that. But it's actually, I thought it was a surprising challenge. I don't know if you guys have run into things like that.
Giselle.
Yeah, I would agree in terms of the regulations and our abilities to be able to do everything we want to do for our patients, in a sense that, like I give the first example about even being able to use the genomic information, but there's still more information, the high throughput drug screening information that we're doing on the research side that still regulatory-wise, if it's not clear, if it's not proven, we can't use in the patient treatment side, where we're seeing high throughput drug screening of our patients' tumor cells and what drugs they might respond to, and being able to correlate that with the genomic information isn't quite there yet. It's there in our minds, but I feel like there's still a lot of regulations that are blocking us for doing, for that patient that's alive right now, we need to be able to have access to that.
All right, we're now to the question part of the session. Yes, would you mind going to the microphone? Thank you very much. Go ahead. And introduce yourself.
Natalia Bukharo. I'm working for The Hive. It's a company that supports translational research by supporting open source translational platforms and tools. So I have a few questions. So my first question is for Kathy. In your presentation, you mentioned different questions, and one question was, whites are different from blacks. And I know one drug that has been approved for African American. It has been a bit controversial, a big deal. But I was wondering, what do you think is going to happen if somebody will try to develop precision medicine for a particular ethnicity? You would think that there will be some big difference, and there are differences between how people, accessibility to disease depends on people's origins. So, but I haven't seen any discussions around precision medicine targeted to people's origin.
It's a really good question. I think what we found by doing the COMPASS study is, a lot of people say it's very difficult to get African Americans into studies. We actually use the right centers, and we were able to get African Americans into the COMPASS study. Our disease skews 2-to-1 African American, and it does not skew hardly at all Asian. So we were able to pull in the data for that. And I think once you start to get the data and understand there are differences, the genomics look different. The African Americans have much less heterogeneity and much less P53 and other things. So when you start to uncover that, and then again, you have to put more money out to say, this is an important thing we want to understand, who wants to work on it, how do we frame the question, then I think you will get success. I think the hard part is getting people to do these kinds of studies are very expensive, and the more you have to keep breaking it down, who's going to pay for that? So that's, I think, part of the challenge.
Okay, and then I have a question for Josh about data sharing. So if I remember correctly, people who are enrolled in clinical trials, they get informed consent that has to be on the 7th grade level. People sharing data is maybe not that intrusive, but there are consequences. There could be consequences, especially with genetic data. How do you make sure that people that share their data actually understand what they're getting themselves into?
Sure. So the informed consent question is really important. In the Seeq for Science work, what we've said is we're working on a technology protocol, and it should work to help patients share their data with research studies or consumer apps or whatever. The informed consent language is particular to any study that's going to use this technology. So for example, in the All of Us research program, which is going to be our pilot study that's using Seeq for Science, there's an ongoing effort to develop the language and the protocols for doing informed consent. And yes, it includes considerations about reading level and literacy and numeracy. It also includes considerations like being able to not just display a block of text, but be able to go back and do a quiz that checks whether people have understood what you've explained to them in the informed consent process. So there's many levels to this. One of the things that we're going to aim to do is to publish the consent language that some successful studies have used and have gotten approved by IRBs. But at the end of the day, it's really a study-specific thing, because the risks and benefits of participating in any particular study are going to be driven by what that study is all about.
Okay, are this data also going to be anonymized?
I apologize. I will take my usual prerogative to say we have to limit questions so that I can hear other people.
My name is John Birch. I'm an angel investor. I'm a member of the Mid-America Angels in Kansas City. And I've been researching healthcare data, and I've got a couple of questions that I suppose David and Josh might be the most relevant, but Kathy, you certainly are going to have some ideas about this. Precision medicine seems to be exposing more and more, just the practice, the potential practice of precision medicine is exposing more and more of the weaknesses of electronic medical records as we know and love them. They exist, after all, as a historical accident of data processing systems that were put in hospitals back in the late 80s, or in the early 80s. And they are, it seems to me, and precision medicine illustrates this better than anything, unfit for purpose going forward. There is a huge growth nowadays, as people may not know, in registry, patient registry software. The market for patient registry software a year or two ago was about $400 million. It's now projected by the end of this decade to be $1.5 billion. That's mainly because of the availability of digital data, because you can just grab it now and do things with it. But it also is addressing, I think, another problem inherent in EHRs, namely the data quality. There's no attention to data quality whatsoever in electronic medical records. So I guess to put this as a question, I'm surprised really at the amount of discussion in these kinds of forums and many, many others that I've been to on the emphasis on extracting data somehow or other from the electronic medical record. In fact, that I would suggest is the wrong way to go. I'd just like to get your comments on that thought.
So I'll just do one example that we were having at lunch today. When you start to look at the precision medicine and genomics, we had done market research across 5 cancers together just last month to see how aware across 5 different cancers were the patients of the importance of their genomic data. And despite some of the best foundations doing endless work in educating patients on genomics, it turned out that the group that would know the most were the ones that actually had drugs because they were genomic targets. So as soon as that advertising happens or patients know clinical trials are opening for a drug that has a certain subtype, that starts to create a big flurry of activity. What we have found is that sometimes it's tricky because if we're starting to develop a precision-based trial, could even be a platform trial or whatever, and we want to accrue all the patients, it's not necessarily that all the genomic information you want is sitting in the patient's record. It's not. And we're not even necessarily asking, we don't want all your genomic data, we just want to know your subtype so we can get you into the right trial. But even that isn't easy to get out of the health record. I don't have any of that data for 20 years, even though I've had extensive genomics done. It's not sitting in my health record on my portal at Dana-Farber today. So we were actually having this discussion that just to do a little pilot, even looking at myeloma, if we have a trial opening and there's 4 different subtypes in that platform trial and we're trying to get it accrued and we need tons of patients for that trial, are we better off doing a direct-to-patient Facebook program, Twitter program, and doing an app that allows the patient to go in and just say to the doctor, check off which of these subtypes I might be? And I think that's giving you an example of how we're trying to understand what we can get from EHRs, because we know it's critically important to get that data. We have lots of clinical questions as well as others. But simultaneously, the world of social media and apps is moving so fast, it may jumpfrog a lot of this, or hopefully they'll just both end up working together. On the registry side, I do think that's critical. I would say of the 5 cancers working together, the one thing that came out of this was we have to have very simple registries that allow us to count the patients. We know who you are, we know you're engaged, we want you to know your subtype so that we can get you to the right spot, the right center, the right trial, whatever it may be at the right time. And that is a huge area of collective focus amongst those cancers.
So I just want to put my thumb on the scale, but Giselle, what do you view as your useful sources of clinical data on your patients? You seem to use registries. Do you use EHR data ever? What's your view on this?
So we combine all of our clinical data in our own database with the genomic data.
How much time do you spend cleaning it up?
Our bioinformatics team spends a lot of time. It's a lot of time.
Yeah.
Yeah.
Good.
So-
Go ahead.
Yeah, I was
David,
gonna-
keep it short.
Okay. Well, I was going to just say then two quick things. So the first is the data are probably absolutely clean for purposes of billing. So the incentives drive what it is that you're cleaning. Harvard Business Review had an article several years ago about actually, when do you clean up data? You clean it up when you have a particular objective and a bottom line. With respect to Seeq for Science and the patient-facing phenomenon that it is part of, if you think about the aspect that can most likely lead to incentivized behavior to clean up the medical record, I'll tell you that it is when you see your own medical record, and you don't need to see it necessarily through a Seeq for Science app. You could just go to a portal, certainly the newer ones, and you can say, my goodness, they have a problem, or they have a list of problems, true for me personally, which are not true anymore. So you have finally the possibility of there being an individual auditor who's incentivized to sort of inform the hospital. It won't solve the problem, but it will elevate it to a level of awareness. And beyond that, it's a longer conversation.
And speaking about longer conversations, I see an eminent researcher and former bone marrow transplant expert/researcher.
Yeah, hi. I apologize for the laryngitis. Save-A-Gyne, I'm at the Farber and at the medical school. And actually, my question is similar to the question that you were just asked, but it does in fact have to do with incentives, really. So where's the incentive to, and where is the money and the effort going to come from to actually deal with the data validation? And that's even without laryngitis, for the data validation part. Because actually, that's only one part of the question, what was just asked. So there's the clinical data, and let me assure you that, that Epic doesn't clean up the clinical data. Because right now, when I do bone marrow on a patient, a child with acute lymphoblastic leukemia, the way that Giselle, Dr. Sholler would, I get three options. If I put in acute lymphoblastic leukemia, it defaults so that you have to enter additional information. That's cool, structured data. The only problem is that I just did a bone marrow to find out what the patient's status is. And the choices are in remission, in relapse, with refractory disease, which I don't know at the time that I have to enter the code. And I cannot go back and change because that's billing, okay? So I am actually entering, by definition, information which is incorrect. I mean, that is actually the grossest example of a fault in the system that I've been able to come up with, because there's no way other than guessing that information can be correct, and then that information is part of the final record. So when you abstract it, or when Shrine abstracts it, it's wrong. If we go to the other side, so who's actually incentivized to check on that? The bills send, if you change it afterwards, by the way, you're in default on Medicaid, so that's a whole different problem. I mean, it's very complicated. If you go to the other side, we act as if the genomic, epigenetic, any other information is correct. I'm a major fan of phenotypic assays. I'm all with you on that. And we know that it isn't. I mean, that's been done to death, Same DNA, divided in two, given to the same postdoc to run in two different samples. The answers are different. It's been done, it's been done, it's been done, it's been reported. So we have data that's not reproducible and not accurate on both sides. So when you talk about the academic-industrial interface, who's actually incentivized to clean it up? And what does it mean to clean it up? And what does it mean to the answers that we get when we use the data?
So I'd like to, because Joe's had a whole week as a CEO to really think in depth about these issues, I'd like to ask him, so you're dealing with this cutting-edge whole genome sequence, and you've indicated that there's going to be a gradual progression in the goals around that sequence. Nonetheless, you do have, even today, a data curation / accuracy / QC problem. What's the answer from your perspective to Eva's question? Where's the incentives? to keep it clean or cleaner?
Yeah, so I mean, I would say that, well, there's definitely incentive on the company side to return correct results to the participant, and then on the genomic data, I'm actually not familiar with the results saying you can split the DNA and get different answers. I could imagine different analyses, but the sequence, I presume, would be, at least to a first approximation, very, very similar. And then on the phenotype side, I'd say that a lot of the incentive comes from the participant themselves, in the sense of seeing, like you mentioned, when I see my medical record and I see an error in it, I'm incentivized to correct that error, whereas in general, my physician.
But what if you don't have a good intuition of what your genome looks like?
Right, on the phenotype side. On the genotype side, I can tell you we get, like people have an intuition for how their ancestry should be, that's for sure. And they tell us all about it. And there are incentives there. But for any individual genetic variant, I mean, I think the only way forward here is it has to be multiple independent validation.
So I don't want to let Ethan off the hook here, since it's good to torture people individually. But we've seen in animal studies, for example, weird things like after the fact contaminants from prokaryotic genomes in mouse genomes, weird switches. How do you reassure your pharmaceutical collaborators, and how do you incent your own team to actually check up on and make sure that you have adequate data quality?
Yeah, I mean, a lot of the places, so, there's either two options. So you're either going into a rare disease where there's a foundation of literature, and sometimes maybe people have made a first generation passive models, and so you've got a benchmark to compare to. So there you can kind of look at literature and say, do we find something consistent? In cases where it's a black box, much like NGLI1, where there was basically one researcher doing most of the work, but was really looking at it, I'd say primarily from an enzymological or biochemical point of view, there it's hard to know. You're truly doing discovery work. And there the check and balance comes from having a community of very active folks, including the advocates who are essentially earning PhDs by virtue of their being steeped in it. And then of course any researchers that are drawn into that field. So that's the best way to get rid of a contaminant is to get it out in the open of a community. And so sharing pre-competitively, sharing is the only way to disinfect the.
On that very positive note, I have to say we have to end because we're actually going late. And so I just want to thank the panel for a really wonderful exposition. Next amazing panel, 2:30. Don't be late.